So, greetings all, and welcome to the Science of Childhood Cancer lecture series that's hosted by the St. Jude Children's Research Hospital Comprehensive Cancer Center. I'm Dr. Charlie Roberts. I am Cancer Center Director at St. Jude, and it's my great pleasure today, along with my co-host, Dr. Elizabeth Fox, to welcome our speaker, Dr. Kathe Janeway. It's a real pleasure for me to be co-hosting Dr. Janeway today because I got to know her quite well when I was in Boston a number of years ago, and it's fantastic to see her career trajectory since then. And so it's a real pleasure to introduce Dr. Janeway. She's Director of Clinical Genomics at Dana-Farber Cancer Institute, a senior physician in the solid tumor program at Dana-Farber Boston Children's, and an associate professor of pediatrics at Harvard Medical School. Educationally, she did her undergraduate at Barnard College at Columbia University, where she was Phi Beta Kappa, then on to Harvard Medical School for her MD degree, Boston Children's for residency, and then Dana-Farber Boston Children's for her fellowship in pediatric oncology. The focus of Dr. Janeway's research is genomics of pediatric cancers, including expanding the benefits of precision oncology to kids with cancer through basket trials and data sharing. In addition, she conducts translational genomic research and clinical trials of new therapies for pediatric sarcomas. It's important, I think, to note that she's co-chair of the Molecular Characterization Protocol Working Group for the NCI's Childhood Cancer Data Initiative, and recently awarded a fellow of ASCO. Dr. Janeway, it's a pleasure to throw it to you.
So much for the invitation to speak and for the kind introduction. I'll just get my screen sharing up here. Great. So what I want to speak to you about today is learning from children and designing clinical trials with their data. So these are my disclosures. And in terms of an outline, I'll first begin with a couple of slides to just present the context of our work, which will include recent results of precision oncology studies showing the clinical impact of molecular tumor profiling in pediatric cancers. I'll then move on to provide you with the interim results of our GAIN Consortium ICAT2 study, which is a precision oncology, a multi-institution precision oncology study that we run here, and then I'll move on to sort of highlight some observations from that work and some of our ongoing studies. So first, to orient everyone, I will focus on pediatric solid malignancies. This is a pie chart of sort of incidence of pediatric cancer, and the solid tumors are this rainbow of what I like to call ultra rare cancers that represent about a third of pediatric malignancies. Sorry, skipped ahead. So one question that we posed, we sort of started to ask ourselves many years ago, is do children with pediatric solid malignancies benefit from molecular tumor profiling? And the reason, sort of context for this question is that in the United States, molecular tumor profiling with targeted next generation sequencing tests, which are usually DNA and less often RNA tests, are standard of care for advanced adult solid malignancies. That's represented by FDA approval of a number of these tests and also inclusion of testing in NCCN guidelines for a number of adult cancers, including, as I'm showing here, for non-small cell lung cancer. The studies that support this practice have mostly not included pediatric solid tumor patients, but nevertheless, some pediatric solid tumor patients have molecular tumor profiling with targeted NGS tests, at least in this country. But we think that the access to that testing is not universal. In addition, there are several recent reports, and I'm showing 3 of the most recent ones here, which have used whole exome sequencing, whole genome sequencing, whole transcriptome, and sometimes methylation arrays to evaluate the genomic landscape of individual patients' tumors, return that information, and then follow the patients and see if they're able to receive matched targeted therapy. These studies, as I'm showing you here, are mostly nationally funded programs or well-resourced institutions. And I'm going to highlight for you, since we're speaking on the St. Jude's seminar series, the Genome for Kids study right here. And what these studies have shown is that it is possible to identify molecular alterations with diagnostic and therapeutic significance, that molecular data clarifies diagnosis in some patients, and that patients receive and respond to molecularly targeted therapy. Similar to these studies, a major effort of my work has been to conduct precision oncology studies. Just to clarify what I mean by that, we mean sort of taking the patient's tumor, evaluating that from a molecular standpoint, identifying genomic alterations that might, where there's evidence for the potential activity of matched targeted therapy, trying to get the patient the drug and then determining what the response is. And our objective here has been, as it is for precision oncology, generally in the studies the published studies I just told you about, to improve outcomes for patients and decrease side effects. But we've aimed to do this to help not only the individual patient, but also to learn from those individual patient experiences to fundamentally change care for young people with cancer. And the way that we've done that is a little bit different than the studies that I just showed you. Because of the context in which we work, we've really focused on targeted next generation sequencing, both DNA and RNA, and I'll get into that in a couple of slides. But the approach that we've taken is to get patients together so that we can observe patterns, identify genomic alterations, make a connection to the drug, get the patient the drug, and measure and record the response. So I'm going to focus on telling you about the GAIN Consortium ICAT2 study, and this is the sort of a schematic of the study design. Patients are eligible if they have a high risk or recurrent refractory childhood solid tumor and are age 30 or younger at the time of diagnosis. Sequencing is performed from both tumor and blood. The tumor tissue, we use sort of archival FFPE samples for the most part. We perform targeted NGS testing and a little bit of whole transcriptome sequencing. We return laboratory test reports and a clinical interpretation to the oncologist. We collect clinical data, including baseline demographics and diagnosis. Obviously, we have the sequencing data, and then follow patients for treatment and response. I'm going to share with you the results of an interim report of this study. Some of this was presented at the 2021 ASCO annual meeting by Alana Church, who's my terrific collaborator in this project. And our purpose for this interim report was to support data-driven insurance coverage decisions and development of testing guidelines in the United States to inform molecular testing practices in clinical settings where resource limitations may prohibit acquisition, storage, and transport of fresh frozen tissue, and comprehensive sequencing, by which I mean sort of the type of sequencing in those other studies I showed you in the introductory slides. So the primary aim of the GAIN Consortium ICAT2 study is to describe overall survival according to whether patients received matched targeted therapy identified by tumor profiling. Secondary and exploratory objectives include describing the frequency and range of molecular alterations, to do in-depth assessment of extraordinary responses to matched targeted therapy. And for this interim analysis, we really focused on describing the molecular alterations identified, characterized with regards to impact on diagnosis, prognosis, or response to matched targeted therapy. And we also looked carefully at the intermediate clinical endpoints to identify extraordinary responders. And we conducted this interim analysis after the first 389 patients were enrolled. So just as I mentioned, I'll tell you a little bit more about the molecular tumor profiling approach taken in this study. So all patients have OncoPanel performed OncoPanel is a targeted NGS DNA assay. It covers introns from almost 500 cancer-associated genes, exons of 500 cancer-associated genes, and then spans the introns of a subset of about 60 genes shown here to be able to identify rearrangements. We then have a sort of selective approach for RNA sequencing. RNA sequencing was initially whole transcriptome sequencing, but now we use a targeted RNA-seq panel designed for solid and brain tumors. And that approach to RNA sequencing basically assumes is divided depending on whether or not we anticipate identification of activating fusions for the particular diagnosis being profiled. So we perform RNA sequencing if there are activating fusions expected that are not targeted by the OncoPanel, then we automatically perform RNA sequencing. Sorry, that's this group in the middle. If we expect an activating fusion or an oncogenic fusion, and the DNA panel does not identify that fusion, then we will proceed to RNA sequencing. So this is sort of at the same time, and this group is done sequentially. One important point I want to make is that these panels, the OncoPanel and the RNA sequencing panel we use, were either designed by pediatric specialists or were designed with pediatric input. In terms of clinical interpretation of the molecular tumor profiling results, this is a very complicated slide to explain something fairly simple. So we created for this study a knowledge base that we call iCatalog. And basically what this allows us to do is we feed in molecular data and a limited set of clinical data, and it allows us both to access previous knowledge from prior clinical interpretation of the molecular tumor profiling, and also to link out to sort of publicly available data on specific genomic alterations. Within the context of this iCatalog, we also create a clinical interpretation report, and that clinical interpretation report addresses both the sort of significance of the identified alteration for diagnostic impact and therapeutic impact, and we also, in the rare cases where it's applicable, discuss prognostic impact. This iCatalog allows us to generate this clinical interpretation report. And so basically the study participating treating oncologist receives both the laboratory report and also this clinical interpretation report. We do have a molecular tumor board, but we only discuss about 25% of the cases at the molecular tumor board because most of the sort of clinical interpretation, either we've already discussed that specific variant and its associated clinical impact at a prior molecular tumor board, or the evidence is very clear and doesn't require discussion. We reserve discussion for sort of unclear evidence or conflicting evidence or topics we haven't previously discussed. In terms of outcomes and measures, we classify our genomic alteration in terms of potential clinical impact using AMP, CAP, ASCO guidelines for diagnosis and prognosis, and ICAT tiers for therapeutic associations. The ICAT tiers were developed for our initial sort of feasibility pilot of this study, with tiers 1 and 2 referring to clinical evidence, linking a particular molecular alteration to response to molecularly targeted therapy. Tiers 3 and 4 referring to preclinical evidence, and tier 5 being associated with consensus opinion. For extraordinary response to match targeted therapy, we needed to develop a definition for this study. We required that the patient be receiving only monotherapy, that they have a RECIST radiographic response, or a duration of therapy for 4 months or greater in the context of having measurable disease. So moving on to the results. this slide shows you the analytic patient population. These patients were enrolled before January 1st of 2019. Of the 389 patients enrolled up to that date, 345 were eligible for analysis. Patients were ineligible if they did not have an OncoPanel result, and there were several reasons for not having an OncoPanel result. For 6% of those patients, it was a technical failure, meaning none of the samples that we were able to require were able to be sequenced. And for 4% of the patients, we actually do allow enrollment onto this study with an outside molecular report. So that's what the other 4% are. The most common diagnoses were actually sarcomas. So about 2/3 of our patients had sarcomas, with the most common diagnosis being osteosarcoma. When we follow this this interim report patient population along, you'll see that at the time that we did this analysis, about 77% of the patients had relapsed or refractory disease, and 23% had new diagnosis, high risk solid tumors, which generally means metastatic disease. When we look at these patients, we were able to make an ICAT recommendation, or should explain what an ICAT recommendation is. So in our terminology in this study, if we identify a molecular alteration through our testing for which there is either the preclinical or clinical evidence for potential response to match targeted therapy, we call that an ICAT recommendation. So in this patient population, we were able to make an ICAT recommendation in about 65% of patients. And then delving into those ICAT recommendations a little bit more, per patient, you can see that most patients had one or two genes that were associated with an ICAT recommendation. And the highest tier of ICAT recommendation per patient was most often tier 3, tier 4, or tier 5. I'll just make one point on this slide, which is that when we evaluated the molecular alterations for clinical impact in the therapeutic arena, we did that both at the time that the clinical interpretation report was issued, and also with updated evidence at the time of this analysis. So that's what the sort of light pink and dark pink bars refer to here. The most common genes associated with tier 1 or 2 ICAT recommendations are shown in this bar graph to the right, as are by colors the types of alterations that we identified. So you can see sort of PIK3CA, BRAF, SMARCB1, PTEN were the most common alterations there. So then we were interested in seeing, what happened to the patients who received ICAT recommendations. And if you look at this group of patients who received ICAT recommendations, we wanted to follow them for receipt of match targeted therapy and then clinical outcome. So there were 200 patients who had adequate data, who received ICAT recommendations and then had adequate sort of clinical follow-up data to allow us to assess whether or not they received match targeted therapy. So this slide focuses on those 200 patients who had an ICAT recommendation, were alive at the time the results were received, and had treatment follow-up data. Those 200 patients were mostly patients with relapsed and refractory disease, but also some patients, 40, with newly diagnosed disease. And if you follow what treatment they received, you can see that 29 of those patients received match targeted therapy. Interestingly, some patients received no treatment. And in the relapsed and refractory group of patients, there were a subset, 20 of those 160 patients, for whom we wouldn't expect them to receive match targeted therapy because it was not feasible for it to be available to them, which would mean that either there wasn't a clinical trial, there wasn't a clinical trial, and there was no FDA approved drug for which a pediatric sort of dosing study had been performed. So basically, if you look at all of this information together of the patients who would have been anticipated to be able to receive match-targeted therapy, about 30% of those patients actually received match-targeted therapy. So to look at more deeply at therapeutic impact, we as I discussed, defined whether or not patients had a response to match-targeted therapy. And then we can look at those patients in a couple of different ways. There were patients who progressed on match-targeted therapy. There were patients who were not eligible to be a responder, and that was either because they received the molecularly targeted therapy with chemotherapy, or because the patient did not have measurable disease at the time that they started. And so of those patients who received match-targeted therapy, I'll focus in now on the responders. So interestingly, these responders, with the exception of one patient, all had a fusion that would sort of activate an oncogene in some way. And all of those patients, all of those patients had either a rare tumor or a sarcoma. So moving on now to the sort of diagnostic impact of these, of the genomic profiling, we found that 61% of the patients in this interim report population had a tier 1 or tier 2 AMP, ASCO, or CAP, according to AMP, ASCO, or CAP guidelines diagnostic impact. We're showing those impacts are shown in pink in the outer ring of this circle, which is arrayed according to diagnosis. And the majority of those findings with diagnostic impact are fusions, which are shown in green in the sort of second circle here. There are 17 patients who had a real-time impact on diagnosis, by which we mean the sort of enrolling diagnosis of these patients into the study was once we had the molecular data back and we were able to integrate that molecular data into the diagnostic picture, sort of ended up with a revised diagnosis. Interestingly, in 5 of those cases, FISH or other non-NGS assays actually failed to detect the alteration. So what's sort of considered gold standard in this world is FISH, but actually can be sort of falsely negative. I think important to point out, and shown in this fusion, this circus plot, is that the fusions that had diagnostic impact are multiple different fusions with multiple different fusion partners, showing sort of the promiscuous nature of rearrangements or oncogene rearrangements in pediatric cancer. So just to summarize the findings from this interim report, 299 of 345 patients, or 87% of patients, had one or more alterations with potential clinical impact. 29 patients received match-targeted therapy. 7 patients receiving match-targeted therapy were responders. All responders except one had treatment matched to a fusion, and 78% of diagnostically significant alterations were fusions. In conclusion, molecular tumor profiling using targeted panel tests designed with pediatric input can have a significant impact on diagnosis and treatment recommendations for young patients with solid tumors. These results emphasize the importance of fusion detection for patients with sarcomas and rare tumors. I didn't talk about the germline findings from this study, but they are similar to previous studies showing about 10% of patients with sort of autosomal dominant cancer predisposition syndromes, and that's already been published, and I'm showing you the PubMed ID here. There are limitations to targeted next generation sequencing. To some extent, you're limited in your ability to detect gene signatures. It is not possible to use targeted NGS to discover novel mechanisms of cancer development in pediatric cancer, or any other cancer for that matter. Because targeted NGS tests are sort of designed and have to exist in that design for some period of time, they can fail to identify sort of emerging diagnostic alterations or therapeutic targets until they're sort of updated for the next updated and validated to incorporate those new findings. If you're interested in learning and discussing more about molecular profiling in pediatric cancers, I want to point out two sessions at the upcoming AACR annual meeting that will focus on this topic. So now I'll move on to the next section of the talk where I'll address sort of two observations from this and other precision oncology studies in pediatric cancer. And really, both of these observations have the overarching theme that unmet needs can be identified by precision oncology studies. And the first I'll focus on is the prioritization of molecularly targeted therapies for clinical trials in pediatric cancer. So I just need to take a little side track here to talk about pediatric drug development. And I'm going to use COVID as kind of just an introduction to that. So this is my initial COVID vaccine card. I do have my booster now, but this I made this slide a little while ago. And you can see that my first vaccine was in December of 2020. These are my children at the time, my children who are 15, at the time of their first COVID vaccine, which was June of 2021. So I think, you know, what our childhood cancer patients have been experiencing with this sort of delay between drug development for adult patients and drug development for younger patients has been felt much more widely now in this COVID era. So there are really two efforts that I want to mention to try to sort of minimize the delays between the development of cancer drugs for adult patients and pediatric patients. The first is that for cancer clinical trials, in many cases, adolescents can be enrolled simultaneously with adults and can receive the same body size adjusted dose administration as is given in adults. And there are a couple of manuscripts discussing this and an FDA guidance. And there are actually incentives in the United States and requirements in Europe for clinical trials in children. The second effort that I want to talk about is the RACE Act. And just to give some background on that, is this study that I was fortunate to be able to be a collaborator on, looking at approvals of pediatric cancer drugs and studies of pediatric cancer drugs in children. And basically what you can see is that the time for FDA approval for pediatric labeling from the time that it was approved for adults is quite long. And one of the reasons that cancer drugs developed for adults sometimes aren't studied in children is that there are exceptions because the sort of indication is diagnosis based, and for the cancer in adults is diagnosis based. And that diagnosis, for example, lung cancer, occurs only rarely in children. And so the company or the industry partner would sort of get a pass that they didn't need to study that drug in children because that diagnosis doesn't occur. But now we know that many drugs that are being developed for cancer are sort of molecularly based. So that brought about the RACE Act, which is a Race for Children Act that updated the Pediatric Research Equity Act, or PREA. And so now what happens is that if a cancer drug is being developed and going for approval for an adult indication, if the molecular target is relevant in pediatric cancers, then drugs seeking FDA approval need to have a pediatric study plan. But in order to be able to sort of enact the RACE Act, the landscape of molecular alterations in pediatric cancers that need new treatment approaches needs to be understood. And that's really sort of the advanced treatment resistant disease and rare cancers. So looking at the GAIN ICAT2 study, this is sort of a figure that arrays the sort of genes in which there were molecular alterations for which we made an ICAT recommendation. And the sort of tier of the ICAT recommendation is shown here, and the number of samples is shown depending on the sort of size of the bubble. And what you can see from this figure is that there are certain genomic alterations like EWSR-FLY1, which is the sort of canonical translocation in Ewing sarcoma, which is common, but for which there really are not going to be existing cancer drugs in the adult space, because this is primarily a pediatric cancer. But at the same time, there are the occurrence of mutations, for example, in the RAS-RAF-MAP kinase pathway in about 12% of our patients that are shared with adult cancers and for which there are molecularly targeted drugs being developed. So to look at this a little bit more closely, I'll turn to the NCI Children's Oncology Group Pediatric Match Trial, which is really led by Will Parsons and Nita Seibel. But as Dr. Roberts mentioned, I'm vice chair of what's called the screening protocol, which sort of evaluates, does molecular tumor profiling to identify actionable mutations of interest, for which there are available phase 2 clinical trial protocols. And I want to just make one point from this study, which is that in the very recent publication of the first 1,000 patients enrolled on this match screening protocol, the RAS pathway alterations were the most common alterations seen. And in fact, the clinical trial subprotocol associated with that, which was selumetinib, was presented at ASCO in 2021 and accrued much more quickly than expected due to the sort of frequency of RAS pathway alterations in pediatric cancer. And what this kind of observation tells us is that agents targeting the RAF-RAS-MAP kinase pathway should really have, according to the RACE Act, pediatric study plans. And in fact, because of this observation, we'll have at the Pediatric Cancer Working Group Town Hall meeting at the AACR annual meeting a presentation on RAS-directed pipeline from Clay Gustafson. So to move on now to the sort of second observation regarding identifying unmet needs through precision oncology studies, I will next talk about identifying cancer types with poor outcome and clinical trial options at recurrence. So I think I mentioned already that for the GAIN ICAT2 study interim report patient population, osteosarcoma was the most common diagnosis. And in fact, we enrolled at the time of the interim report, 64 patients with osteosarcoma. I thought at first, maybe this is just because I'm an osteosarcoma physician. And so perhaps we accrued more osteosarcoma patients than we might have. But in fact, the pediatric match trial also has as its number one diagnosis osteosarcoma, enrolling 184 patients onto the screening protocol with osteosarcoma. So what is osteosarcoma? Osteosarcoma is the most common bone tumor. It's still incredibly rare. It occurs in somewhere between 600 and 800 adolescent young adults patients in the United States. It's most commonly occurs in the bones around the knee joint, but also can occur anywhere in the skeleton. The disease has a propensity to metastasize to the lungs, which I'm showing you here, and that happens in about 25% of patients. It is a disease of adolescents and young adults. The peak incidence is around 13 years of age for girls and a little bit older for boys, 14 or 15. Again, the disease presents as a localized disease, meaning only the primary bone tumor is present in about 75% of patients. And in this context, the event-free survival, the long-term event-free survival, is only about 60%, with long-term overall survival being around 70%. 25% of patients present with metastatic disease, and in this context, the event-free survival is only about 25%, with the overall survival being about 30%. The genomics of osteosarcoma are quite complex. It is a disease that is characterized in almost 100% of cases by chromothripsis, which is a gene shattering event. Also mutations tend to be, and copy number changes tend to be concentrated in areas of rearrangement that result from the chromothripsis event. That pattern of concentrated mutations is called contagious. Osteosarcoma has had no change in outcome and no new standard of care therapies for the initially diagnosed patient population in 40 years. It does not respond to immune checkpoint inhibitors. There have been no biologic subgroups identified that are associated with treatment response or resistance. And so to sort of subgroup patients to determine their sort of outcome, we look at whether or not patients are metastatic at the time of diagnosis. And 12 weeks into therapy, we look at the primary tumor, which has to be removed or resected. And we look at how many of the cells have died or with chemotherapy, what we call percent necrosis. I want to point out that these two features that we use, metastasis and necrosis, are really observations of the cancer behavior, and they're not sort of fundamental biologic aspects of the disease. And so we can't use those to sort of help us identify new therapeutic avenues for the highest risk patients. And there are no genomically informed disease-specific clinical trials, or there weren't until the trial that I'm going to tell you about in a few minutes. So I was fortunate to work with a then resident, now fellow at UCSF, Amanda Marinoff, who trained here for her residency program, to analyze some of our targeted NGS sequencing data from osteosarcoma patients enrolled into the GAIN study that I just told you about, and also a similar institutional study that we call PROFILE. And we had almost 100 patients. This is a high-risk patient population because of the GAIN study sort of preferentially enrolling higher risk patient population or relapsed and refractory patient population. So you can see that 33% of the patients had metastatic disease at diagnosis, and a larger proportion than you might expect had what we call poor necrosis, or less than 90% necrosis at the time of that tumor resection. This is an oncoprint showing the sort of most common genomic alterations in osteosarcoma in this patient population, and I want to focus first on signature 3. So we identified signature 3 in about 30% of these patients. So I'm just going to take one slide to give you some background on DNA damage response and inhibitors. This figure from a recent review on DNA damage response and inhibitors shows the sort of machinery that's involved in repairing double-strand breaks in DNA, single and double-strand breaks in DNA, and some of the inhibitors that sort of work in this arena. What is sort of the original understanding of this machinery comes from understanding BRCA1 and BRCA2 germline mutations in initially breast cancer and now ovarian cancer. And when there are germline mutations coupled with loss of heterozygosity of the second normal allele in breast cancer, what happens is that there's sort of abnormal repair of this DNA damage that has an over-reliance on PARP. And so then the cancer becomes significantly susceptible to PARP inhibitors. One of the effects of having this abnormal DNA repair machinery is what we call signature 3, which is a pattern of changes in the DNA that really represent a scar from this defective DNA response. So we decided, based on the observation in our data and also in several other published reports, including one of the sort of pediatric pan-cancer analyses from St. Jude, that there's signature 3 in 20 to 55% of osteosarcomas, that it would be important to do a clinical trial of a genomically informed clinical trial of olaparib in relapsed and refractory osteosarcoma. But we were hesitant to do a monotherapy trial because osteosarcoma is a fairly aggressive disease, and we had concerns about relying on just one targeted therapy. So this slide sort of covers the rationale for this phase 2 trial of olaparib plus the ATR inhibitor ceralasertib in patients with recurrent and refractory osteosarcoma. The first bullet point I pretty much reviewed already, which is that we see signature 3 in 20 to 50% of patients. Osteosarcoma patients also have an enrichment of germline mutations in DNA repair genes. ATR inhibitors inhibit the growth of osteosarcoma models. That was initially thought to be related to the fact that they have alternative lengthening of telomeres, and alternative lengthening of telomeres uses much of the same DNA repair machinery, but I think subsequent studies, it's a little less clear that there's a direct association between those two things. PARP inhibitors and ATR inhibitors show synergy in preclinical studies. Those are not osteosarcoma specific, and tolerability in clinical trials. And we thought that a multi-institution PI-initiated clinical trial in recurrent osteosarcoma would be feasible, even though it's a rare disease. I'll go on to tell you why that is. So I just need to tell you a little bit about clinical trial design in recurrent osteosarcoma. And this is yet another example of using data for trial design. So a couple of years back, wearing my hat as the vice chair at that time for the Children's Oncology Group Bone Tumor Committee, we were sort of frustrated by the lack of new agents in this disease. And we thought we need to really test a bunch of new drugs in patients with relapsed and refractory osteosarcoma. And so we need a trial design where we can get some preliminary evidence from a clinical trial for a new drug to sort of bring forward in this disease. But we needed an osteosarcoma specific trial design for a couple of reasons. The first is that RECIST response, which is the usual phase 2 clinical trial endpoint in relapsed and refractory cancer, is not an appropriate endpoint in osteosarcoma. The tumors are calcified, they have a calcified matrix. And even in the setting of newly diagnosed patients receiving chemotherapy that's proven to improve overall survival, we really don't see RECIST responses. And the second is that 25% of our patients with relapsed and refractory disease have lung-only pulmonary nodules that can be removed with surgery. And it is standard of care to remove those lung-only pulmonary nodules. So 25% of our patients don't have any cancer in their body at the time that we're looking to treat their relapsed and refractory disease. And those patients aren't eligible for standard phase 2 trials that use RECIST response. So what we did to come up with this osteosarcoma specific trial design is we looked at the data from patients who had enrolled in previous COG Children's Oncology Group phase 2 trials studying new agents. We had about 96 patients who had measurable disease at the time of trial entry. And we looked at their event-free survival. And what we saw is that it was reproducibly terrible. So patients with measurable disease with osteosarcoma, when they enter a phase 2 trial, 12% of those patients, 88% of those patients have progressed at 4 months, or only 12% remain progression-free. And the confidence interval around that 12% is very narrow. It's from 6 to 19%. For patients who have completely resected disease, we looked at a single prior Children's Oncology Group trial that had enrolled about 45 patients. And here the outcome is not as bad, and it doesn't become as predictable until later after trial enrollment at about 12 months. So at about 12 months, 80% of patients have progressed, or 20% are progression free. So we said, because we have these very reliable benchmarks, you can use those in clinical trial design. And we were able to successfully do that in a series of children's oncology group phase 2 trials that followed that, some of which were led by the bone tumor committee and some of which were led by our early phase group or developmental therapeutics group, which actually Dr. Fox is part of the leadership team of. And basically what we were able to do here was evaluate a number of new drugs. Unfortunately, none of those met our bar of activity, so they were all negative trials, but they did all enroll quite quickly and give us the answer to whether or not these drugs were of interest in osteosarcoma quite quickly. So that allowed us to think, well, we could actually have a trial that we initiated studying olaparib in combination with ceralasertib. And I'll tell you a little bit about that clinical trial. I am showing you the clinical trial number here if you want to look up more information. So patients are eligible if they have recurrent osteosarcoma, are 12 to 30 years of age, and 40 kilograms or greater. We will enroll patients from both of these groups of recurrent osteosarcoma patients that we see in the clinic. The cohort 1 patients have measurable disease, and the cohort 2 patients have disease that is planned to be removed by surgery and can be fully removed and is restricted only to the lungs. The primary objective of this study is to look in cohort 1 to determine whether olaparib in combination with ceralasertib improves the 4-month event-free rate in patients with recurrent measurable osteosarcoma as compared to that benchmark based on his historical COG experience I just showed you. For cohort 2, what we're interested in is estimating the proportion of patients who have paired pre and post treatment tumor samples that can be submitted for correlative studies. Treatment on this study, we're really happy to be working with oral agents. So both of these drugs are given orally and the cycles are 28 days. The endpoints we're using are disease control at completion of 4 cycles of therapy for cohort 1 and submission of those paired samples for cohort 2. In terms of statistical design, we're looking to see a 38% progression-free rate at 4 months, and that is a significant improvement over the upper 95% confidence interval of the historical population, which has 19% rate. This study has a 2-stage design with a total enrollment of 38 patients, with 15 of those patients being enrolled in stage 1. So just the status of this clinical trial, it's currently being amended to update dosing based on results of some trials in adults. We're anticipating that we'll reopen to enrollment in June of 2022, and we're open at Dana-Farber and 3 other clinical trial sites. Importantly, we admit that we don't understand everything there is to know about DNA damage repair in osteosarcoma, and so we're partnered with the Sweet Cordero Lab and have a very tight relationship between sort of correlative biology work in his laboratory and the clinical trial. So going back to this study of osteosarcoma conducted by Amanda Marinoff, I'm going to focus now on MYC amplification, which is seen in about 12% of osteosarcomas in this study and in other studies that use NGS that have been published recently. And I'm only going to show you one figure, but just to say that MYC amplification, maybe not surprisingly, in this patient population is associated with a worse outcome, even though this is a high-risk patient population. And we were able to validate this finding in a second patient cohort that used a different approach to copy number analysis. So we have some next steps with this MYC amplification study. We need to validate the results in a uniform national patient population, and that's underway with children's oncology group samples. In the context of doing that, we will need to determine what the appropriate copy number cutoff is, assuming that we replicate the result. We also need to associate MYC amplification with clinical features and prognostic factors, clinical prognostic factors. That relationship is not yet determined in this study or the one other recent publication on this topic. In order to do that, we need a large patient population. If we're able to gather that large patient population that has appropriate sort of genomic characterization and associated annotation with treatment and outcome factors and baseline clinical features, that may also allow us to identify other molecular subtypes. So to accomplish this, we are incredibly fortunate to be working with Corey Painter and Nick Wagley, who established a sort of concept of conducting genomics research called Count Me In. And they established that initially with a project called the Angiosarcoma Project. And what this concept does, or what Count Me In does, is to really bring the research to the patient. So our sort of traditional approach to research is to bring the patient to the brick and mortar kind of oncology provider institution or the hospital. But more and more people access information, and during COVID, even their doctors, through sort of their cellular telephone or their computer. And so the idea here was really to bring the research to the patient, and not only that, but to partner with the patients in sort of designing the research. And so for the Angiosarcoma Project, which is an incredibly rare sarcoma, they were able to engage 640 angiosarcoma patients across the country. Through analyzing samples submitted through this project, we're able, and the sort of associated medical record data, they were able to identify a clinically important subtype of angiosarcoma. So we sort of right before the pandemic, we partnered with Count Me In and osteosarcoma patients and advocates to found the Osteosarcoma Project, which launched in February of 2020 and already has 257 registered patients who've submitted 82 saliva samples for sequencing. We were able to successfully get a Cancer Moonshot grant, which is part of and become part of this patient engagement and cancer genome sequencing project, which will allow us to relaunch the Osteosarcoma Project with what we're calling shared learnings, which is basically for patients who send in tumor sample, we're going to do that, sequence those tumor samples on a CLIA approved platform and share back sort of what we're learning about osteosarcoma from the sequencing of that patient's individual tumor. We'll also be able to provide the opportunity for patients who opt in to get germline testing with return of results. And we're hoping to relaunch very, very soon. The protocol is undergoing final revisions and resubmission to the IRB. So in conclusion, molecular tumor profiling has benefit in pediatric malignancies, solid malignancies, and fusion detection is particularly important. It's really essential to continue to generate molecular data, both for clinical care and also discovery, and efforts to combine data between institutions and of different types will be essential for further understanding of childhood and other rare cancers. And annotating that molecular data with deep phenotype and longitudinal clinical and outcome data will be essential. So with that, I'd like to acknowledge my research group and collaborators and funders. Thanks so much.
Great. Well, thank you so much, Dr. Janeway. That was truly an amazing talk. And on behalf of myself, Dr. Roberts, the entire St. Jude Science of Childhood Cancer team, and the more than 130 participants in today's lecture, I really want to thank you not only for today's lecture, but for taking us from a place in the early days of precision oncology in pediatric cancer now to what you just showed us, which is bringing it directly to the patients and getting their input on how this impacts them directly. Truly an achievement. I see that Dr. Roberts is with us. Charlie, comments from your side? I know there are also some questions in the chat we want to get to.
No, I just think Dr. Janeway, fantastic talk, fantastic work. It's great to see the progress and how much we're learning and how illuminating of still how far we have to go. Unfortunately, we're still going to be employed for a while, but it's you
Yeah.
who I think are helping lead the way in the next wave of our understandings and precision therapy. It's just great.
Thank you. Well, it's been, well, I look forward to the discussion.
Terrific. We have about 3 minutes to the top of the hour. Dr. Janeway has graciously agreed to stay on a little bit longer for questions, and I do have a couple in the chat, but I know that many may have to drop off in the next couple of minutes. So I do want to take a moment to announce our next Science of Childhood Cancer lecture, which is next week, April the 7th, in the same time slot, which is the pathobiology of asparaginase resistance by Dr. Alejandro Gutierrez from the Dana-Farber. So let's get back to discussions with Dr. Janeway at this point. I'm going to open with a couple of questions that are in the chat while others come in. Dr. Roberts, in the early part of your talk, while you were talking about ICAT-1, had a question about your molecular tumor board and the fact that you discussed 25% of the cases there. And how frequently did those discussions establish or alter the diagnosis or treatment plan?
Yeah, so for the molecular tumor board discussions, in terms of the diagnosis, we altered the diagnosis in 17 patients. So 17 of 345 patients. So that's about 5%. But for those, and those we worked really carefully to be sure that we really understood everything that was known about the patient as they came into the study before we did our molecular testing, right? We wanted to make sure we didn't miss anything. So and that's been described in pretty much all of those published studies that I showed you, where molecular approaches are used. And that is a solid tumor specific number. I just want to point that out. I think in brain tumors, CNS brain tumors, actually, molecular profiling is now part of the WHO criteria for assigning diagnosis. So it's actually probably even more important in brain tumors. In terms of the way that the molecular tumor board works in our study, it's maybe a little bit different than in some other studies. So we tend to, so if we make a decision, for example, that, we're going to recommend an ICAT recommendation for MEK inhibitors and BRAF fusion cancers is a tier 1 or tier 2, that there's clinical evidence for that. We don't necessarily rediscuss that for every single case. We wait until there's some new evidence. If the new evidence concurs with what we've previously done, we just continue to just issue our clinical reports without bringing that to the molecular tumor board. So the molecular tumor board is used more for sort of difficult situations where there's conflicting evidence regarding a relationship between a genomic event and response to molecularly targeted therapy. For the sort of the proportion of patients who had a therapeutic recommendation is about 60%. So where we found a molecular alteration where either an FDA approved drug or a clinical trial might be considered based on the molecular findings, that number is around 60% in solid tumors.
Terrific, Katie. And that actually sets up nicely for the next question, which is regarding the response rate in the ICAT study. And, we know there are multiple steps to getting to the response, but we wondered if you could reflect a little bit on the contributing factors to that. Specifically, the question was, is it about physician education and adherence to recommendations? Do we need better drugs? Are there other things that you can think of that impact that?
Yeah, so that's a great, so there are two questions in that question. So the first question is, what proportion of patients actually received matched targeted therapy? And then the other part of that is sort of the response rate to matched targeted therapy. So I'll talk first about the response rate to molecularly targeted therapy. I think it's going to be important to start to look at combination therapies for pediatric cancer, or using match targeted therapy sort of much earlier in the treatment course, as is done or as is being studied right now for leukemia or neuroblastoma. That I think is going to be incredibly important. In terms of patients actually receiving match targeted therapy, that is very complex. So that has to do with clinical decision making in the, between the doctor and the patient. Many of our relapsed pediatric solid tumors have sort of standard chemotherapy, and I think pediatric oncologists tend to use those before they go to clinical trials or sort of novel drugs, because response rates are high, even if cure rates are not high.
Terrific. Other questions, again, I encourage people to put questions into the chat, because Dr. Janeway, again, is willing to stay on and answer some more questions. Questions currently in the chat. Dr. Culp says, amazing talk, Dr. Janeway, and asked the question, how does precision oncology deal with intratumoral heterogeneity? And then there are a couple other follow-on questions, but I'll let you start there.
Yeah, that's a great question. So we haven't, the bottom line is we haven't figured out how to deal with that yet. We discuss it often. So one of the common discussions in our molecular tumor board is we find a particular genomic alteration that we think is, where there's good evidence for response to match targeted therapy, but the allelic fraction is only 5%. What do we think about that? And the answer is we really don't know. I think it's going to require a lot more of the studies like this, and a lot more sort of data aggregation to understand how to think about tumor heterogeneity. And in fact, we haven't really even figured this out in adult cancers yet in precision oncology. And I think we'll learn a lot as time goes on from adult cancers. I think using circulating tumor DNA is going to be important in the tumor heterogeneity field as well.
Terrific. And then are you able to track or distinguish between driver and passenger alterations, or track whether the alterations occurred in cancer stem cells or more differentiated cancer cells or metastatic sites?
Yeah, again, this is an excellent question. The methods that we use certainly do not do that. I think people are developing methods to do that in more sort of experimental context, not so much in the sort of clinical return of results context, that hopefully will come eventually to the clinic.
And next question is, are there efforts, whether they be clinical trials or other research efforts at the Dana-Farber or elsewhere, to use CRISPR therapeutics to treat osteosarcoma and other cancers?
Yeah, that's a really good question. Too bad Dr. Roberts isn't here still, who knows more about CRISPR than I do. I think people are definitely thinking about CRISPR therapeutics. There's no question. Right now, CRISPR is mostly being used to identify what we call therapeutic vulnerabilities. In other words, in particular cancer types or in particular molecular contexts, does that create a dependence on certain genes or certain sets of genes where that we can then use or leverage that information to sort of bring new therapies to the clinic? So right now, CRISPR is an amazing tool in the laboratory, but may eventually be a therapeutic, just not yet.
Great, and I can say that we still have more than half of the participants are with us participating in this question and answer session. Again, please put questions into the chat. While those are coming through, Katie, can you reflect a little bit on, from your perspective and career, the learnings from those early days of precision oncology, and how you think about keeping pace in the clinical trial space with the fast pace of identification and better understanding of molecular targets?
Yeah, it's a great question, Beth. So I think You know, one of the interesting developments in pediatric oncology, and I think the RACE Act is really sort of helping this, is that we are more and more sort of approached to do clinical trials in pediatric patients, in particular, sort of molecular subgroups. And you know, figuring out a way to sort of make sure that every patient has a molecular profile that they sort of carry with them, that pretty soon after their diagnosis, is something that I think is sort of the next challenge for our field. Because having that information will help us in so many ways. First of all, it helps us, the people, I'm using that us very generally, not so much about me, the people who design these early phase clinical trials, our industry partners in those trials, know sort of how feasible clinical trials are, what the designs need to look like. So that's one piece that it helps us with. The other is that for the patient and the doctor in the clinic, sort of when a relapse happens, if you have that molecular information already, it will really help you think about what the options are and to sort of compare our sort of standard of care next step for chemotherapy versus whatever molecularly targeted therapies are available. And I think that will help with decision making sort of earlier on in the relapse refractory sort of disease course. I'm not sure I quite answered your question, but one thing I feel like we've learned is that waiting until patients have, you know, quite advanced disease to understand what the molecular makeup of their tumor is maybe not the best approach for precision oncology, especially as we enter this era, hopefully, where we'll have a lot of trial opportunities for patients.
Great. There's a detailed question in the chat that I'm actually going to paraphrase and make a little bit more general. And it's about something you touched on about clinical decision making. And I'll paraphrase the question to be, you know, as you think about clinical decision making and the vast array of really disparate types of therapy that are out there, how do you guide patients, say with osteosarcoma, through the, you know, immunotherapies, the targeted therapies, the cytotoxics? How do you think through that?
Yeah, no, that's a great question. So I think that typically the way pediatric oncologists think, I'll go back to kind of the theme I was on in the last question. We typically think for initial therapy at the time of diagnosis, right? We want to choose the treatment plan that clinical trials have shown us to date is going to produce the highest likelihood of cure. Okay, so that's the initial selection. If there's a clinical trial ongoing that allows someone to get either that standard of care or something that we're researching to find out whether it's better, worse, or the same, then we opt for that clinical trial. Usually we offer it, at least. We want to give people the opportunity to participate. So that's sort of the newly diagnosed decision-making paradigm. And actually, I'll just want to make this point again. In some cancer types, we're far enough along in precision oncology that some of those clinical trial opportunities now are adding precision medicine to our standard of care treatment, which is really important to make that point. I already mentioned neuroblastoma does that. Leukemia has a trial like that. Some of the brain tumors are starting to have those kinds of trials. So then when we get to, if that initial treatment unfortunately does not work and we're in now the relapse and refractory setting, what has been kind of the go-to for a long time in pediatric oncology and oncology in general is sort of our standard second line chemotherapy. That's typically what we've done is we've said, you know, we have knowledge from previous studies that this next kind of chemotherapy, you know, will be active in this situation. Unfortunately, those active chemotherapies, cure rates after them are usually quite low. And so I think what I was trying to get at in the last answer is that what I hope is that going forward at that time when we have, if we have to sit down with a relapse refractory situation and talk about that next line of treatment, it's really at that point that we should be understanding what the molecular makeup of a cancer is so that we can incorporate precision oncology trials into that decision making at that time. So, you know, that is really the next decision. And those tend to be sort of phase 2 trials that we're thinking about at that time versus a sort of standard second line chemotherapy. And again, hopefully more and more of those phase 2 trials will have a molecular component for pediatric patients. And then, you know, later on, if those initial approaches don't work, then we usually are looking at brand new cancer drugs. And at the current time, that's mostly when we're considering molecular alterations, is to looking at those brand new cancer drugs. And I think that will continue. But hopefully we can move some of that consideration up into that very first time point of decision of, you know, should we try the standard chemotherapy or should we try maybe standard chemotherapy plus a molecularly targeted therapy?
Thanks. That was a fantastic and really comprehensive answer about, you know, thought process in this very complicated world. I'm not seeing any more questions in the chat. I'm going to give folks on the line, because there's still quite a few folks listening in, a moment or two to put those questions in. And while we give that little pause, Katie, I want to thank you not only for participating in this lecture series, but truly for being able to present a landscape and the contributions not only that you personally have made, your institution, but through collaborations with cooperative groups and really bringing thought processes and trial design to childhood cancer and precision oncology. We are getting thanks, Dr. Katie, for an awesome, inspiring talk, and other kudos in the chat. But Katie, we can end here, but I do want to give you the opportunity. Are there questions we should have asked you that we didn't, or any other parting thoughts you want to leave us with?
I wasn't expecting that question. That came from right field. So let's see. The most common questions I get, I think were covered, sort of the heterogeneity question. You know, why do so few patients get molecularly targeted therapy, and why is what can we do to make the response rates higher? Those are really the questions that people tend to ask. I think really my only parting thoughts are, there's one thing I want to bring up that I wasn't able to bring up, which is there's actually an effort underway in the United States called the Molecular Protocol, which is through the Children's Oncology Group to really try to start to do some of that molecular tumor profiling more at the time of diagnosis. It's not available yet for all cancer types, but it is starting to be available for some of the brain tumors. And I think there's going to be efforts to expand it to some soft tissue sarcomas as well. So I wanted to mention that because I didn't have a chance to talk about that. And I want to do some shameless advertising again for the AACR annual meeting. If you want to learn more about that molecular protocol I just talked about, or some of the other precision oncology studies that are recently published, then there's sessions on Monday and Tuesday that we would love to see you at. And the Sunday night pediatric cancer working group town hall will have 3 speakers who are going to talk about really interesting sort of new therapy areas in pediatric oncology.
Great. Well, thank you so much, Dr. Janeway, and we look forward to seeing you at AACR and in the future. And again, thanks for joining us for the Science of Childhood Cancer, and thanks to all the participants.
Thank you for the invitation, and thanks for listening.